Okay guys, in this brief video we're going to talk about one of the ways that DNA can be proofed, especially after replication, in order to find mistakes and get those fixed quickly. The specific proofreading mechanism we're going to talk about is called excision repair. Excision repair is outlined here in this graphic on the right, but basically it has 4 different steps. First of all, let's start here at number 1. You have a template strand of DNA. In the process of DNA replication, DNA polymerase, remember, reads one base pair at a time. Suppose that within DNA replication that the polymerase put in the wrong base pair. What's happening here at this next level is outlined by what's called a lesion. If DNA polymerase put down the wrong base pair, meaning the wrong nucleotide to match the one complementary to it, it will create a lesion. It will bubble out because the hydrogen bond cannot form. And actually, if you have unmatched nucleotides, then they can repel each other and create a bubble in the DNA. So now we have what is called a nuclease. A nuclease is an enzyme that fixes these lesions in the DNA. So again, imagine DNA polymerase came through, laid down some base pairs, but it laid down the wrong base pair in this case, created a lesion. A nuclease comes in and clips out that mistake. When that mistake in DNA goes away, now you've created a gap. The gap that's left over has to be filled with more DNA. So the only enzyme that's going to fill the gap is, of course, DNA polymerase, which is an enzyme that makes a polymer of DNA. And DNA polymerase goes back over it, and this time you've sealed the gap using another type of enzyme called DNA ligase. It's an enzyme that binds together DNA. So as a result of excision repair, we have several different enzymes at work. We have a DNA polymerase, which led to the problem to begin with, and also fixes the problem by laying down a new set of complementary base pairs. DNA ligase binds together the new strands of DNA that were created after the proofreading, and a nuclease is an enzyme that clips out mistakes in the DNA. So how frequently does DNA get mutated? What is the mutation rate or mistake rate of DNA? It's actually very, very small. 1 in 10 billion nucleotides results in some sort of mistake that would lead to a lesion and lead to excision repair. So it's actually a very low amount. But this is one of the processes in which DNA that becomes initially damaged can be repaired so that these damaged pieces don't get carried through to other parts of the central dogma if proteins are ultimately to be made.